Welcome to Botany class, Chapter 3, Enzymes, Part 1, Chemical Reactions. In this we will discuss about chemical reactions of enzymes. First, we will discuss almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These are called ribosomes. Example, 23S or RNA. Here, enzyme as a primary structure contain protein. Example, amino acid sequence of the protein. An enzyme, like any protein, has a tertiary and a secondary structure also. If we observe tertiary structure, you will notice that backbone of the protein chain folds upon itself. The chain which crisscrosses itself. Hence, many crevices or pockets are made. One such pocket is the active site. This active site of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzymes through their active site catalysis reactions at a high rate. Enzyme catalysts differ from inorganic catalysts and organic catalysts. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperature above 40 degrees centigrade. Enzymes also isolated from organisms which normally live under extremely high temperatures. Example, hot vents and sulfur springs. Stable and retain their catalytic power even at high temperatures up to 80 degrees to 90 degrees centigrade. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. Here, in chemical reactions, chemical compounds undergo two types of changes. One is physical change. Simply refers to a change in the shape without breaking of bonds. This is a physical process. Another physical process is a change in the state of matter. Example, ice melts into water when water becomes a vapor, and also physical process. Here, when bonds are broken and new bonds are formed during this transformation, it is called as a chemical reaction. Here one example, barium hydroxide which react with the sulfuric acid H2SO4 which gives barium sulfate and water molecules. In this inorganic chemical reaction, another one is organic chemical reaction. Example, hydrolysis of starch into glucose. Here, the rate of physical or a chemical process refers to the amount of product formed per unit time. It can be expressed rate equal to P by T. It is called a velocity if the direction is specified. Rates of physical and chemical process are influenced by temperature among other factors. General rule of thumb, rate doubles or decreases by half of for every 10 degrees centigrade change in either direction. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed ones. When enzyme catalyzed reaction are observed, the rate is vastly higher than same but catalyzed reaction. Example, in this we will observe. Suppose carbon dioxide plus water. Action of carbonic anhydrase enzyme. It convert into carbonic acid H2CO3. Here, if any absence of enzyme, reaction is very slow without 200 molecules of H2CO3 being formed in an hour. So by using the enzyme present with the cytoplasm, it is called as carbonic anhydrase. The reaction speed dramatically without 600,000 molecules being formed every second. The enzyme accelerates the reaction rate by about 10 million times. The power of enzyme is incredible indeed. There are many types of enzymes present. Here, catalyzing a unique chemical or metabolic reaction is different. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called as metabolic pathway. Example, glucose plus oxygen gives rise to 2-pyruvic acid plus H2O molecules. Let us discuss how do enzymes bring about such higher rates of chemical conversions. Here we know already active site. The chemical or metabolic conversion refer to a reaction. The chemical which is converted into product is called as a substrate. Hence, enzymes are protein with a 3-dimensional structures including an active site. Convert a substrate into a product. So, substrate is indicated with the S, product is indicated with the P. Here, S has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is obligatory formation of ES complex, and this complex formation is a transit phenomena. During the state, substrate is bound to enzyme active site. A new structure of the substrate is called as a transition state or structure is formed. After bond breaking or a making is completed, the product is released from the active site. The structure of substrate gets transformed into the structure of products. The pathway of this transformation must go through the so-called transition state structure. Here we will discuss altered structural states between the stable substrate and the product. Stability is something related to energy status of molecule structure. Here we can observe a graph. The y-axis represents the potential energy content. X-axis represents the progression of the structural transformation or a state through the transition state. There is an energy level difference between S and P. If P is at a lower level than S, it is an exothermic reaction. It needs to supply energy by heating in order to form the product. It is an exothermic or a spontaneous reaction. Here there is a opposite mechanism, that is higher level of a P and lower level of S energy required heating energy. This reaction is called as endothermic. S has to go through much higher state to transition state. The difference between average energy content of S and that is a transition state is called as activation energy. Enzymes eventually bring down this energy barrier, make the transition state S to P more easy.